In humans, sex determination really depends on the presence or absence of the Y chromosome. It doesn't really matter how many X chromosomes you have. As long as you have at least one Y chromosome, genetically you're considered to be a male. And so we say the Y chromosome determines the maleness. Now, interestingly, there have been a few very recent studies ongoing to determine the minimal number of genes that are needed on the Y chromosome to determine this maleness. And the last that I heard, it's only about maybe 5 genes that are absolutely required for this process. Now, that's not to say that the rest of the genes on the Y chromosome don't have their own essential functions for various things. However, the maleness aspect of it, really just very few number of genes that are required for that. And so what happens very early in development, by about maybe 5 weeks of gestation, embryos potentially hermaphroditic. We have the genital ridges are going to form, and at this point the tissues could potentially develop to form either the male or the female gonads. And at this point they think that this Y chromosome genes, essential genes, start to kick in. By about the 12th week or so of fetal development, primary oocytes can be detected in females. And by about the 25th week, all of the oocytes become arrested in meiosis, and they're going to stay there and they're going to remain dormant until puberty. So this is pretty important. So in utero, all of these primary oocytes are being produced in the females, and so anything that the mother may be exposed to could potentially affect her future grandchildren, which are in her developing daughter at that point. In males, on the other hand, primary spermatocytes not produced until puberty is reached. And so the long-ranging effects of this not quite as well understood. We do sometimes see changes in chromosome number in humans. We covered this briefly when we talked about non-disjunction. 4 of the most common would be, in terms of the sex chromosome changes, that is, would be Klinefelter syndrome. And this is genetically considered to be 47XXY. And so these individuals have an extra X chromosome. It is possible to also see a 48XXXY, a 48XXYY, a 49XXXXY, and so on. And of course, the more chromosomes that the individual has, the more affected that these individuals will become. Turner syndrome happens in females. It is females who only have one X chromosome, and so they're genetically 45 chromosomes, and the X is short. The majority of individuals with this do die in utero, and so if you're looking at sheer numbers of individuals with this, you're going to find many more males with this, and the reason why, again, is because they're just not going to survive. Another change in the chromosome number that we see pretty frequently, relatively frequently, are females that have 3 X chromosomes, and so genetically this would be 47XXX. These are female, and we call it triplo X. They have a very highly variable syndrome. Many of the women who have this are perfectly normal, and they are unaware of their abnormality until they may need a genotyping for something else. On the other hand, there can be females that can be very strongly affected by this. You may also potentially see a 48 with 4 X's or tetra X, a 49 with 5 X chromosomes, that would be a penta X, and as before, the more of these chromosomes that you have, the more severe the phenotype. Males who are XYY, we call it 47XYY, they are usually very tall, over 6 feet. And again, it's pretty variable syndrome. However, there has been a known and somewhat strong link between this chromosome composition and criminal behavior. It may be that males who have this just have more, some of the sex hormones like testosterone and androgens produced, and they just may be more aggressive overall. The ratio of males to females in humans, we assume when we're doing our calculations for different things that the ratio of males to females is 1:1. However, the actual ratio of males to females is not 1:1 in humans. In general, it's closer to about maybe 106 males for every 100 females. And the actual ratio may be even more different if you include embryos and fetuses that were recovered from miscarriages. the predominant number of these happen to be male, and so this skews this even further. The actual ratio between races, it also changes for unknown reasons. Even within individuals of the same cultural background, say everybody in the United States, between individuals who are, say, Caucasian background versus African American versus Asian, there are actual differences between those races, and we don't really know why that is. Between other countries, some of the race differences do make sense, and we'll talk more about that later, and that would be due to things like selective abortion and so on. So one of the reasons why they think that the ratio of males to females is higher is because the Y chromosome is smaller than the X chromosome. Since it's smaller, it has less mass, it may be able to move more freely, may be more motile than some of the X-bearing sperm. So because females have 2 copies of the X chromosome and males only have 1 copy, we have what's known as dosage compensation for X-linked genes. This is a normal process, normal physiological process. What happens is one of the X chromosomes is largely inactivated in normal females, and this helps to equalize the expression of these X-linked genes between the sexes. The inactivated X chromosome we term as bar bodies. So the lion hypothesis is that the X chromosome that becomes inactivated is randomly chosen, and we can see this using the example of calico cats. So in a male who happens to have this orange coat color on their X chromosome, the cats are going to be an orange color. And males who have a B, on recessive black on these particular X chromosome would be a black cat. In the females, if they were homozygous for the black color, it doesn't matter which of the X chromosomes was inactivated, all of the cats would be black. In females that were homozygous for the orange color, it wouldn't matter which of the X chromosomes was inactivated, and the result would be an overall orange color. However, if the females are heterozygous on their X chromosomes, what we can see is through this pattern of random inactivation, in some of the cells, the orange color is going to be inactivated and the black is expressed. In others, the orange is expressed and the black color is inactivated. And so what we see is a very patchy, sort of calico cat colored result. Another way that we will see sex determination is through temperature variation, and this happens in reptiles and some other species. And in this case, it's a temperature-dependent sex determination instead of a genotypic sex determination. So, depending on what temperature the organism, the eggs, are incubated at, you may see differences in the ratio of males to females. And you can see this 3 different ways. And looking on the left here is percent female, the temperature increasing along the bottom. In some cases, if the temperature is cool, they're going to be primarily all females, and as the temperature increases, they're going to be primarily all males until you reach this real sweet spot where 50% would be males and 50% would be females. Conversely, you can have cases where low temperatures are going to be primarily male and the higher temperatures primarily female. You can have other cases where there might be 2 ideal temperatures. Very high temperatures, you're going to have primarily females. Moderate temperatures, you're going to have primarily males. And again, higher temperatures, you're going to have primarily females. And one of the big problems or issues with this is if we look at things like climate change. If indeed the Earth is getting warmer by several degrees, this does not bode well for many of these species. Imagine if the temperature is increasing, they're going to be either all females or all males. Well, you're not really going to have a very successful second generation if that is the case.